Hello, everyone. So, today I'm going to be talking about the formal analysis of biological circuits using high order logic theorem proving. My name is Usman Hassan, and I'm an associate professor at the National University of Sciences and Technology in Pakistan. My co authors are Professor Syed Abid, who is from Kuwait University, and then we have Adnan Rashid, who is my postdoctoral fellow at the National University of Sciences and Technology in Pakistan. So we have organized the talk in this manner. So we will start with some introduction about synthetic biology and the motivation for having a more rigorous and accurate way of analyzing these circuits and controllers. Then we will present the proposed methodology. This is going to be followed by the summary of some of the formalization results that we have obtained so far, and then finally we conclude while presenting the summary of the work as well as describing some future directions that we are pursuing at the moment. So synthetic biology is actually the process in which we apply engineering principles for analyzing biological systems with the motivation of having more efficient analysis. So mainly we are concerned with electrical systems, control systems, and computer science based foundations, and they are applied on various aspects of biology, including biological circuits, pathways, and controllers. There are numerous applications for synthetic biology, including bioenergy, aquaculture, environment, agriculture, and healthcare. So there's a lot of interest in these kind of analysis. Um, so mainly the synthetic biology analysis is based on two components, which are biological circuits and biocontrollers. Biological circuits actually allow us to capture the parts of a cell which mimic the logical functionality that is performed by electrical circuits. So it's very close to electrical circuits. And then we have biological controllers that are used to regulate the functionality of different modules and components of the overall system. So mainly these are the essential components of synthetic biology. Now, the whole analysis process can be broken down into three main steps. And the first step is to capture the dynamics of the given system that you have while representing the interaction of its modules and subcomponents using differential equations. So you get a set of differential equations. And once we have captured the behavior in these set of differential equations, the next step is to extract the transfer function that represents the dynamics exhibited by these differential equations in the frequency domain. And in this process, we usually utilize the block diagram representations that are very commonly used in control system analysis. And finally, we use Laplace transform theory to perform the transfer function based analysis of these systems, and this way we can obtain the desired properties of interest of the given system that we are dealing with. Traditionally, these analyses are done using paper and pencil proof methods, and the main idea is that you create the differential equations or write them on a piece of paper, and then once you have those mathematically represented differential equations, then you can use Laplace transforms and block diagram representations to analyze the whole system based on mathematical reasoning on a piece of paper. Now, this is very suitable for smaller systems, especially because the analysis is based on mathematical foundations and it is supposed to be accurate. But as we start to deal with larger systems, the whole process becomes quite human error prone because we as humans can make mistakes while doing the analysis, and especially when dealing with large systems, this is going to be more probable. So that is why people have started to use computer-based analysis tools, and numerical simulations really tops that list. The idea is that you construct a mathematical model in a computer, and then you use different sampling-based analysis techniques to find the correctness of your properties. But since the analysis is usually done on based on sampling, so that is why the results can not be categorized as complete. Moreover, the algorithms that are involved in the whole analysis process are not formally verified. So we do not really know that these algorithms are going to be doing what they're supposed to do. So they may mess up the whole analysis. So this is one of the other main limitation of numerical simulations. So we can conclude that none of these existing techniques offer an accurate and complete analysis. And given the safety critical nature of synthetic biology and its applications, it's very important to have a technique that allows us to achieve accuracy and completeness. So basically, in this work, we propose to use theorem proving, which is a formal methods technique, and given its soundness, it guarantees to provide accurate and complete results. The main idea is that we develop a computer-based mathematical model of the given system, and then we use deductive reasoning to find if a given property holds for the given model or not. So the technique has been used for verifying various hardware and software systems, as well as mathematical theories have been formalized using hydrologic and theorem proving. So we want to use the same principles um, to analyze systems in the synthetic biology domain. And we also want to use the whole light theorem prover in this context. The main motivation behind this choice is the extensive support that this theorem prover provides for multivariate calculus, including complex numbers, integrals, differentials, and Laplace transforms, because these are the essential components of the analysis that we have at hand in this work. Now, this is our proposed methodology. As already described, it is composed of three main blocks. You have to construct the model of the dynamical behavior of the given circuit. You have to capture its diagram representation, and then you have to work with the transfer function. Now, in order to provide support for these things within a hierologic theorem prover, we first of all we built on the existing support on multivariate calculus as well as Laplace transform, and we develop a formal model, as well as we also develop theories that allow us to represent block diagrams as well as continuous dynamical models within the framework of a hierologic theorem prover. So with these extensions, which are the core of the presented work, we can not only construct the formal models, but we can also verify the desired properties that involve transfer function for the given systems and circuits within the sound core of a theorem prover, and this way we can get the verified block diagrams, we can get the solutions of the dynamical models, as well as we can get the verified transfer functions. And since the whole verification is done within the sound framework of a theorem prover, the results are guaranteed to be correct. They are guaranteed to have all the all the assumptions that are required to for the analysis to hold. Now, the main components or the main focus of this particular paper that we are presenting is on the formal modeling of block diagrams of any generic biological circuits, as well as we want to build support to find out the transfer function of any biological circuit, and the technique that we use behind it is mainly inspired from the block diagrams that are used in the context of control systems in electrical engineering. So we have four configurations. We start with the first one, which is the configuration that has a series of components. So we want to find the net transfer function of a series of components. And as you can see in the figure in front of you guys, that there are two components connected in series, and the net transfer function of these two components which are connected in series is the product of the individual transfer functions. So we actually model this behavior using a hierologic function which is called series comp, which is given here. It accepts a list of transfer functions representing the individual transfer functions of the submodules that we have in the series connection, and then it returns the product of the individual transfer functions of the given blocks. Similarly, we have the second configuration, which represents the parallel connection of components, and the behavior that we have is that we just add or sum all the transfer functions to get the net transfer function of this configuration. Again, we developed a function which is called sum junction. It takes a list of transfer function as components which represent the individual um, transfer functions of the blocks which are connected in parallel, and returns their corresponding sum. Then we have the third configuration, which is composed of a block which is followed by a parallel blocks, which are again their transfer functions are there, and then we can find the net transfer function as described here. And again, we defined a hydrologic function that is called pick point. It accepts the transfer function of the individual block that is upfront, and then the list of transfer functions which have and their transfer functions are represented in this list, and then we get the net corresponding transfer function as described here. And finally, we come up with the fourth configuration, which is the feedback block representation. This is one of the most widely used configurations, both in the control theory as well as in the context of synthetic biology. Now, in order to formalize this one, we actually utilize the series block. And as you can see that as we go along this feedback path, it's actually composed of a number of series blocks through which we go through. So that is why if you look at our formal definition, it utilizes the series function, and then there is the initial block, and then you have the summation of the branch which captures the behavior of the series, and then we go to infinity because this loop is an infinite loop that goes all the way and all the time. And then this is how we represented the feedback block using our formalization of the previous blocks. So this was the first component, and the second component was the Laplace transform theory. We have already formalized all the properties of Laplace transform, as well as we have formalized it in its traditional way, as you can see it in front of the slide. This is the mathematical definition of Laplace transform, and this is how it is formalized using our definitions. As I said, it really follows through the mathematical definition. We also formalize its conditions and all of its properties like linearity and Laplace of a differential equation and so on and so forth. This can be, more details can be found in our papers. Finally, we also formalize the differential equation. As you can see, the function diff equation n order actually allows us to model differential equation of any order. It takes n, which is the order of the differential equation, then the list of the components, list of the coefficients, and then you have the function which needs to be differentiated, and then the variable that you want to use for differentiation. So now let's come to the main focus of our case study, which is on genetic circuits. Now, gene expression is a technique that allows us to transmit information from passive DNA to the active proteins. Now, this technique is mainly composed of two steps, and the first step is called transcription, in which a section of the DNA is read out into an RNA, and then the translation step comes in which we convert a short strand of the transcribed RNA into the protein. Now, this process is usually regulated in synthetic systems by transcription factors, and there are two transcription factors. The first one is called activator, that increases the transcription rate, and the second one is called repressor, that inhibits the transcription. So basically in this paper, as a case study, we formally analyzed the genetic circuits of both the activated and the repressed expressions of the protein. Now, this is the first case, which is the activated expression of the protein. It describes the interaction of the incoming activating transcription function A with its promoter and the regulation of the expression of gene Y, producing the protein Y, as you can see in the slide. This is regulated by the following differential equation. And this is our formal definition of that equation. As you can see, we utilize the differential equation order n function. The first one is of order 1 and the second one is of order 0. And so we have captured the dynamical aspects of this activated expression of protein. Now we also model the block diagram corresponding to the activated expression of the protein as follows. And we utilize the series component function for that. As you can see that the blocks are connected in series in this case. Once we have modeled these two definitions, now we can proceed to the verification within the sound core of a theorem prover. And as you can see that we verified the corresponding transfer function based on the dynamical behavior that we had captured, and we also um, utilized the dynamical model to verify the same transfer function. And a similar exercise was also done for the repressed expression of protein, and these are the dynamical models and the transfer functions, and more details about the verification can be found in the paper. Now, um, one of the main limitations of our approach was that we are using a higher order logic theorem prover for the analysis, even though the analyses are correct. But the problem is that due to the undecidability of higher order logic, we had to interact with the theorem prover and guide it through the whole process, which involves a lot of human guidance. And this kind of human guidance is usually not expected to be provided by biological experts. So, in order to facilitate the whole analysis, we developed a tactic that is called the TF-TAC, and it allows us to automatically verify the transfer functions of biological circuits. More details about this tactic are provided in the paper, and this really allows us to automatically verify these theorems, and which makes it very convenient for biological experts to benefit from our formalization. Now, finally, to conclude the work, basically the paper is about a higher logic theorem proving based framework for analyzing dynamical behavior of biological circuits. The main support that we provided is composed of the formalization of the block diagram representations of biological circuits, and this formalization is basically inspired from the control theory that is used in electrical engineering. Moreover, we also provided the support for formal analysis of activated and repressed expressions of the protein. Now, there are many other avenues that we want to explore in this domain. First of all, we want to formally verify some more control theory properties of the biological circuits, including sensitivity and stability, and this would allow us to further expand the scope of our analysis. Moreover, we also want to formally verify more genetic circuits and their associated controllers, including auto-activation of gene, phase lag, and lead controllers, genetic phase lag controller in feedback with a single gene as well. And again, the main intent is to expand the scope of our formalized analysis so that a variety of synthetic biology problems can be analyzed within the sound core of a theorem prover. So this brings me to the end of the talk. Thank you very much for your attention. If you have a question or you have any comments, please feel free to write to us. Our coordinates are given on the slide. And once again, thank you very much.